want to start with just a brief introduction. You might have noticed that there is a name change to our educational webinar series that we host every month. It used to be the IHCC International 100K Cohort Consortium Educational Webinar. Many of you may know that we have a sister organization called the Global Genomic Medicine Consortium that concentrates on the implementation of genomic medicine around the world. Along with that consortium, the IHCC and the G2MC sit under an umbrella, a nonprofit company called the Global Genomic Medicine Collaborative. And that is GGMC. You'll see it on the title of Cindy's first slide, and you'll see our little emblem up in the corner there that says Global Genomic Medicine Collaborative. That's a lot of different acronyms, but just to orient you, it's still the same webinar series. We've just expanded our scope out to cover all of our members and our other consortium who are very interested in what we talk about in IHCC. And I think we'll learn a bit about implementation of genomic medicine from some of the speakers that we have lined up, that Latif has lined up coming forward. So let me quickly tell you about our speaker today. Cindy Lawley is passionate about innovation to make multiomic technologies easier and cheaper to measure and to lower the barrier of entry for implementing scalable, reproducible solutions, advancing individualized medicine and health equity. Cindy, in over 14 years at Illumina, and together with her customers, has helped develop solutions to advance the understanding of genetic risk and holds several Excellence in Technology Transfer Awards for her work leading Illumina's Consortia program. Cindy joined Olink in 2020, soon after the launch of the Explore platform. And the Explore platform leverages adaptation of a next generation sequencing technology as a high throughput proteomic readout. She'll explain more about that during her talk, I'm sure. Cindy drives population health initiatives within Olink, working closely with the UK Biobank, FinGen, Scalable, and other large cohort studies like our membership that's tuned in today. So Cindy, we welcome you and look forward to your talk. We'll do about 35 minutes, however long it takes, and please save your questions for the end, and we can do that by chat, or you can be on video, however you'd like to ask your questions.
Sounds great. Great. So thanks so much for that intro. I am so honored to be here to speak to this audience. I do work for a commercial organization and have, as Scott said, worked for several. So I was certainly at Illumina for 14 years. But what I'm focusing on today is really what's excited me about my career transition and what I've seen moving us toward implementation of genomic medicine in the future. One of the things that really struck me, you know, when you give presentations, you can always just hope that you create one memory. And one of the memories that Eric Topol created for me was this sense that it takes on average about 20 years to move something from research to the clinic. And I hope that genomics will change that average, because I've seen some amazing implementation of genomic tools over the course of my career. And I hope that some of the work that I'm doing now will also help accelerate that and bring down that time frame or that average. So it gives you some context of what I care about. And I'll talk a little bit now about my history and my transition to the work that I do today. And then I'll spend most of the talk talking about the work that I'm doing today. So sometimes you get lucky. And sometimes you're in the right place at the right time. And when I came out of my PhD at UC San Diego, I met John Stolpnagel, who was one of the founders of Illumina, because he was looking for a project manager to help deliver data using the array genotyping technology that Illumina was known for at the time, but not very well known for. So Illumina was a pretty small company, about 150 people. There was no sequencing technology at that time. And in fact, when I started in 2004, the only product that Illumina had out in the market was this array matrix format that is in the slide here along the continuum around 2004. After I joined, those arrays that I think people understand are useful, particularly for characterizing GWAS studies at a low cost. Those array tools were being developed when I was in that project management position. Some of my early customers were like Decode Genetics and 23andMe, and I had no idea that the era we were going through at the time, which I think was an amazing catalog of human diversity. So like I said, sometimes you get lucky and you learn a lot, and you get kind of attached to that pace of learning. So what I thought was really remarkable in looking back at that era was the way that Illumina developed content on arrays to get broader and broader coverage, right? We at the time put out a gene-centric array, sort of to support candidate gene approaches. And very quickly it became clear that tag SNP approaches and trying to cover a broad coverage of the genome was going to be a more well-received approach. And so we went from 300,000 with the HAP 300K to 550,000 up to 1 million, and then eventually up to 5 million, shown here. Then what happened was, once diversity was better understood across lots of populations, and this included through sequencing. So here I'm putting the arrow for when Solexa was acquired by Illumina. And that Solexa technology, of course, really transformed how people thought about sequencing. And then eventually it transformed the cost quite dramatically to where we are today. But that collection of diversity was essential to understand how the diversity was related to each other, to understand how the SNPs were related to each other, and then to sort of contract the size of those SNP chips down to, I think 23andMe uses one that's less than 1 million today. Some of the more popular arrays are around that size, that are really low cost to run. And so this reflecting back on that era, I think has some implications for what we're seeing in proteomics today. So I'll move along and tell a little bit more about my history here and what I care about, and then we'll start talking about proteomics, or multiomics, really. So back then, around 2014, we built what was called the multi-ethnic genotyping array. And this was a step change in our understanding of African diaspora, and African genomes really were not well characterized at all. But African diaspora was through 2 different consortia. One was the Page Consortia and Emer Kinney's team, and the other was the Kappa Consortium that was Kathleen Barnes' team. And then, of course, we, over time, were able to leverage the content from sequencing from those 2 consortia, and I've highlighted these in red here, to build that multi-ethnic genotyping array. Or the mega chip that Illumina still sells today, as last I've heard. So using that content allowed us, the royal us, the scientific community, to understand, to run more samples, to genotype more samples, understand better the relationship among SNPs within those genomes that were more diverse, that we were all hoping to integrate into those genotyping chips, and the tagging SNPs characterizing that African diaspora specific genetic variation. Was this era was one of the things I did at Illumina that I'm most proud of. And it really was about being at Illumina, but really taking care of customers that were telling us what was needed to have better equity in healthcare in the future. So this GWAS era, they continue to increase in size and scope. And of course, GWAS, the need for power in GWAS has influenced the collection of these large biobank projects like the UK Biobank and FinGen and other many of these IHCC initiatives that are part of IHCC today, but we're still lagging way behind in diversity. So I guess this I include in here just to say this is, I really thought we were improving on this back in 2014. I think we've kind of been stagnant. And I think that I'd like us not to fall behind in understanding proteomic diversity the way we have in genomic diversity. And so I'm grateful for initiatives that are prioritizing the equity, enabling equity in healthcare. So despite that, certainly lots of diseases have been cataloged against the chromosomes, the human chromosomes. Here's the EBI catalog as of about 1.5 years ago. And this image I want you to hold in mind, because we are cataloging sort of proteomic diversity in the context of genomic diversity in much the same way. So some of the maps that are being generated with proteomic or PQTLs, proteomic quantitative trait loci, look very similar to this image. And so I'm drawing some parallels between those. And then, of course, understanding this diversity allows us to now try to characterize polygenic risk scores, because as we all know, these diseases don't have smoking guns. One single variant responsible for the penetrance or the risk of a disease is pretty unusual. We had hoped for more diseases that had a clear genetic responsibility, but in fact, it's the contribution of many different genes that influence, many different variants that influence risk in most common diseases. And here I'm just cataloging that rise in publications around polygenic risk. And I think there's a lot to be thought about in terms of what genes are contributing to our risk in the genetic space, and how does that show up in the proteomic space. So that's sort of my laying the groundwork for what I'm doing today. And I've spent lots of years working at a company that was trying to drive down costs of collecting DNA and RNA information, RNA being really the technology. That RNA sequencing was really the technology in those early days of Solexa under the Illumina umbrella that sold those sequencers, the exciting ability to move to a digital gene expression profile to characterize real world data. Very exciting. But from what I'm seeing, the RNA agrees with the proteins only about 30 to 40% of the time, often, and there's good biological reasons for that. And so I have been very excited about the ability for proteins to be measured directly in ways that complement our existing approaches to measure proteins directly, and those existing and gold standard approaches, of course, being mass spec, ELISA, those methods. The traditional mass spec methods have not allowed us to easily see low abundant proteins in large numbers of samples. And so that had remained a bit of a challenge. And I think that's where Olink really shines in complementarity to mass spec approaches. So being able to have a view of these protein signatures, I think, is really important, because most, 95% of the clinical trial targets are proteins. And most of the over-the-counter and prescription medications that we take are targeting proteins. So these are really business molecules of the cell, and I'm excited about getting a little closer to phenotype by measuring proteins in the technology that Olink provides. So now I'll talk a little bit about that technology. And I'm really trying to focus on the science and where I think we are today, and really trying to focus on the educational aspect and the opportunity for speaking to all of you. So again, thank you so much for being here. So how does this work? This proximity extension assay, which is the assay that Olink has been founded on, and Olink was founded in 2016. The reason it's innovative is that it allows for you to have very, very high specificity, but in very low volumes. So again, seeing these low abundant proteins and being able to see them consistently in large numbers of samples. So what we do is we have two antibodies, and this is a video loop that goes back around, but we have two antibodies that target each protein. And attached to each of those antibodies are single-stranded oligos that can then hybridize to each other. You can see it here. They can then be extended. And then it's like any other library prep where you can amplify that signal up and count the representation of the proteins in their original sample. But you can do it on a qPCR instrument, which was our primary instrument from 2016 until 2020. And in 2020, I saw them launch, Olink at the time, launch the NGS readout and the ability to use a NovaSeq 6000. And that really excited me. I had known people trying to develop proteomic platforms when I was at Illumina, and just had never seen one that had the ability to reliably read out large numbers of samples, in this case, 384 samples per NovaSeq run. And currently we're at about 3,000 proteins, with the announcement that we're trying to broaden our coverage of the proteome. So that's the technology. And I hinted at this, but I'll just emphasize it. So this graph shows 1 dot per protein in that first library that launched on the NGS sequencer. This was in 2020, and this is when I approached Olink and I joined Olink. So 1 dot per protein from the highest abundance in the upper left corner, or upper left quadrant, to the lowest abundance in the lower right quadrant. And what you might notice here are the density of the dots in this lower right quadrant. What you might also see is this LDL and HDL. These are discoveries that have already made it to the clinic, right? But down in the lower right quadrant, we have the opportunity to make discoveries that are not well understood yet, because those proteins are hard to measure using our traditional technologies, certainly in large numbers of samples where we have lots of power. So I think that's the exciting aspect of this technology. And then the peer-reviewed publications, I think, are a testament to the fact that we are really shining a light on something that hasn't been easy to see. We've got over 1,200 peer-reviewed publications just since 2016. So that's I think pretty exciting. Here are some of the biobanks and cohorts that we've worked with, particularly within a project called Scallop, which I'll talk about in a minute. But first, let's talk about the UK Biobank project. This is a pharma proteomics project where pharma partners came together to cover the costs of running proteomics on, originally it was around 53,000 samples in the UK Biobank. It's now a little more than that, because 3 additional partners joined once technologies were reviewed, and Olink was chosen based upon the features that the group of pharma partners wanted to see out of the data. So very excited about the data being publicly available in this pharma proteomics project. So I really want to emphasize that. And the first preprint for the first half of the data on this UKB PPP project, again, over 50,000 samples in around 1,500 proteins. That's the first data set. There will be a second data set doubling those proteins, but these data are currently available publicly. And they have stated the goals in this preprint, so I just highlight them here, and it's 3 for 1. So we hear this often from customers, particularly in the pharma space, where they're able to have a systematic approach to identifying potential therapeutic targets. They have the opportunity to identify novel biomarkers, and then they're able to understand the biological mechanism underlying each of these. So really understanding the biological mechanism of genetic discoveries. And that is super exciting to me, to layer on and add value to what we've already learned in genetics and this massive investment in genetics. So now I want to talk about so the UK Biobank, that's the preprint, an early publication where they just looked at correlations between, let me actually go back. They've just looked at correlations between the genetic data and the protein data. So these protein quantitative trait loci, they've reported over 10,000. So here, 10,248, and 85% of these were novel. And again, this is just on 1,500 proteins. So they should have the full publication on all proteins out in a peer-reviewed publication. But this just documents that diversity and those associations. Scallop is a powerful precursor to that UK Biobank Pharma Proteomics project. Scallop is an effort that is run through Pfizer and Karolinska Institute, so a co-affiliation, and has combined data from over 40 cohorts and over 70,000 samples. So I'm going to talk now about how Scallop really laid the groundwork, and other publications did as well, but this is the example I chose, laid the groundwork to what was possible by layering proteomic data onto these genomic resources. So this is the milestone publication. Remember I said there were 70,000 individuals represented in SCALLOP today. This is just 30,000 individuals, just over 30,000 individuals, and they just looked at 90 proteins in this project. So remember the UKB, PPP has almost 3,000 proteins. So just in 90 proteins and 30,000 individuals. Now, here's that image that I told you to look out for. They were able to map onto the chromosomes these associations between genetic variants and protein levels. And what this really just means is there appears to be a dosage effect, but we don't know that it's causal. And so these have been mapped across these chromosomes, and this is a figure out of the publication. And what they were able to do is then layer on 38 different diseases and traits onto these data, do Mendelian randomization, and narrow these 450 pQTLs down to 25 that appear causal. Of those 25 that appear causal, I've got two buckets shown here, the blue circle and the green circle. 14 were known therapeutic targets, so that corroborates that their effort is actually showing something that we already knew. And excitingly, 11 new potential therapeutic targets. So this is the structure within which they might have the potential to map potential therapeutic targets that pharma companies can then prioritize or deprioritize. Again, this is a figure out of the paper. The purple box is emphasizing potential repurposing opportunities or opportunities to know in advance potential safety concerns with drugs. And so these are actually drugs that target pathways identified in the analysis where they might be considered for new diseases or new indications. So how does this look? I just mapped it on chromosome 1, how some of the examples look. I just grabbed chromosome 1 because it's a nice big chromosome with lots of examples on it. And so a validated finding among those 14 validated findings was at IL-6R, and this is targeting this protein, IL-6RA. And this is a known therapeutic target that is targeted by tocilizumab, which is used in rheumatoid arthritis. It's developed by Genentech. And then novel target that was identified from the analysis on chromosome 1 was for F3, which codes for a TF protein. And that relationship was with HbA1c levels, so potential for metabolic disease. And then some potential repurposing opportunities that mapped to this chromosome as well are shown here. And I'll just emphasize this one at IL-6R. So that the idea here might be that tocilizumab could be considered in asthma and atrial fibrillation. Now, whether that's a good idea or not, that's up to the pharma company to evaluate. And then the eczema has an asterisk because the directionality of the relationship with eczema suggested that this might be a safety concern for a treatment that targets this pathway. And indeed, tocilizumab lists eczema as a potential side effect in using that medication. So hopefully this helps characterize sort of why these might be useful and why 13 pharma partners might be really keen to layer proteomic data onto the genomic resources like the UK Biobank represents. And then I thought I would do you all a disservice if I didn't share the results of interviews that I've done with, let me quickly say publicly available resources, and then I'll go on to interviews that I did around how that pre-competitive collaboration was successful. So that'll be my next slide. This one is just emphasizing webinar replays. There was a webinar on April 26th highlighting the resource on the UK Biobank Research Analysis Platform. So again, freely available data for approved studies that apply for use and access of those resources. Freely available. Really it's the UK Biobank Research Analysis Platform that has that structure, and that's supported by DNA Nexus. And then the full summary statistics. So this is the relationship between genetics and protein levels that are already done, so people don't have to do these associations themselves. So these are the PQTL analyses. Those are loaded on the Synapse website that I have shown here, and I can share these slides, of course. And big thanks to Matthias Arnold who ran those analyses and loaded those up into that portal. So freely available resources. And then here's my slide around the how, when, and why this pre-competitive collaboration of 13 pharma partners was so successful. So here I just interviewed folks and I said, how is it that this was able to be done? Because this is one of the number one questions that's asked by people talking to those pharma partners. How do we have pharma work with us to use our valuable biobanking resources? And so what they've told me, and I'll just summarize these really quickly, is that the why these pharma would come together. Some of that I've talked about in the slides previously, but it's often the ability to get fast access to innovative technology is only possible with a large buying consortium like the pharma partners represent. This allows, sort of a really quick advance in our understanding, in this case, understandings of proteins and therapeutic targets and biomarkers that they were interested in. The middle pane is intended to emphasize the when. So their messaging to us was that it was very important to, on the heels of previous consortia, in this case, it was the exome sequencing consortium and the sequencing consortium that had already happened with the UK Biobank. And actually those data are still coming out from that sequencing consortium. That was a great time where those partners were already together, where new technologies could be considered and they could work with each other to understand the value of what those might represent. And then the third pane, I think, is probably one of the most important and something that I hadn't thought about characterizing before. But what they all said to me was it was really important that the pharma partners were seen not as a funding body, but these are very accomplished scientists within pharma that were able to work together to characterize the value within each of their own companies to gain support for being able to move this pre-competitive collaboration forward. So I think those are wonderful lessons that I just wanted to capture and share broadly. And I can talk in more detail to those if anyone has questions. So then the next thing I want to talk about is that you don't need 50,000 samples to add value by layering proteomics. And it's a biomarker study in oncology. This study only had 79 samples. So this is from a cohort called the Aravale cohort. The Aravale cohort is a healthy cohort of around just under 5,000 individuals that was collected over time with 6-month intervals of plasma collections prior to cancer diagnosis. So they had 10 individuals that had biobanked 3 samples at least in the cohort prior to their cancer diagnosis. So for those 10, they found matched controls in that database and did longitudinal protein measurements for those controls. And I love this image because it shows the controls down at the bottom, these gray dots. So all the dots are measurements, protein measurements. And then this shows the 3 out of 10 samples that had cancer, and this is breast, lung, and pancreatic cancer. The colors orange, blue, and green. And these were the 3 out of 10 individuals who had metastasized cancer, so stage 4 cancers. The other 7 out of the study were not metastasized. What this showed was that CEACAM5, this is just one protein out of the proteins that they measured, and they measured over 1,100 in this study. It demonstrated potentially a tissue agnostic or cancer agnostic signature for metastatic cancer. Now, this is a preliminary result, certainly needs to be corroborated, but this was, I thought, one of the most exciting aspects of this study. I'll make one other comment about this purple line that maybe you can see here. That was actually one of the control samples, as I understand it from reading the paper and talking to the authors. It was expected for it to gather with the controls, but in fact, it had an elevated level of CEACAM5. And so they reached back out to that individual to find out if that individual had any diagnoses, and they were not able to reach that person. But they did see that in their record that they had what was a benign skin cancer removed, a melanoma removed. So I thought it was great that they included that in the paper. despite not really having closure on that. And then this is an oncology paper that is one of many that fit in that early detection bucket on the left side here. Here I'm just showing a few of those oncology publications that really I see classifying in early detection, disease progression or understanding of mechanistic biology of oncology, and then treatment response. Like, how do we classify and stratify individuals as to how likely they are to respond to treatment? So again, this was a biomarker study that had a relatively small sample size. These publications are, that biomarker study is this one right here that says scientific reports, the one from Magis et al. That I just presented. But all of these publications can be leveraged through a free resource that Olink provides called the Insight app. So this is a web-based application, and this slide shows that Insight app. So anyone can make a free account within Insight and browse proteins, see what some of the signatures are that are present in certain diseases, and see normal ranges in a healthy cohort, as well as other data stories like that for COVID-19. It's my favorite place to convert gene names over to protein names, because genes have many different names, whereas proteins or uniprot IDs are very specific. So very exciting. And then there's a cancer story here around 12 different cancers and the signatures that are relevant in those 12 different cancers. So this is a data story that you can see within that Insight app. Here's the area under the curve for those 12 different cancers and their signatures and their ability to be characterized. I'm sorry, I'm having to quickly scramble and plug in my laptop because I didn't do that in advance. But these show that these proteins were under 100 proteins were able to characterize these 12 different cancers with an area under the curve of 0.93. I'm just going to grab my plug in really quickly, sorry. And then this begs the question, well, why not take these I think it's 85 total proteins. Why not take these proteins and build something that can be taken to the clinic? And indeed, we have customers that are very keen to move some of these disease-specific signatures to the clinic. And so I'll show a few examples of those customers that have some exciting results that they are characterizing for clinical utility. These are two examples. We've got a dozen different customers that we're working with to support them in their efforts to characterize disease-specific multi-analyte signatures. In this case, it's Octave Biosciences in multiple sclerosis and the Crohn's and Colitis Foundation in inflammatory bowel disease. I like these two examples side by side simply because they both have a funding round that allows them to actually move this with funding to clinical utility. And that's certainly what it takes. They're important diseases where diagnosis can happen quite early in life. Multiple sclerosis, often people are diagnosed in their 20s, very expensive, the healthcare system. And then the IBD, this was actually a pediatric cohort. The Crohn's and Colitis Foundation characterized a protein signature using AI. And this IBD signature, of course, with a child being diagnosed, is going to be extremely expensive to the healthcare system. So very important conditions to have better solutions for. On the left, the goal of the panel in multiple sclerosis is to monitor disease progression as part of a dashboard as these patients move from one therapy to another. On the right, the Crohn's and Colitis Foundation tool is for being able to predict the likelihood that a child might have complications from their IBD diagnosis in order to maybe insert a pause before removing a child's bowel, before allowing for time for trying TNF-alpha therapies or other therapies. Because once you take out a bowel, it's really hard to go back. Impossible, obviously. And then for each of these, there's a round of funding from Octave to take this to the clinic. They're actually, I think, at 48 million last I saw. And then Crohn's and Colitis Foundation, which is a nonprofit, now has this IBD Ventures arm to bring in bring in funding to take this to the clinic. And so with that, I'll just go back to this original slide around, sometimes you get lucky and you're in the right place at the right time. And certainly when I was at Illumina, I was very lucky coming straight out of my PhD into a company that had so much for me to learn. I think the proteomics, I approached Olink, and the culture here as a Swedish company is just second to none. It's magnificent. So bringing good culture together with great technology is my dream, and I'm having the time of my life. One of the things I'm really excited about is the context of polygenic risk and proteomic risk, and how this comparison between what's in our genes and what's in our proteins might change over the course of our lifetime. And I think there's lots of things to discover here. And then the last thing I'll say is that SCALLOP, which I mentioned earlier, it also has a clinical trial arm with the aims of enhancing insights and improving power to combine studies, particularly clinical studies, but they certainly have academic partners. And I just wanted to put a shout out to them. And with that, I will take questions. Thank you so much for your attention.
Cindy, thank you so much for a wonderful tour de force of both the technology of Olink as well as the partnerships and the consortiums you've been involved in, applying that technology to life-saving applications. I believe it's very exciting.
Thanks, Scott.
Questions? Yeah, thanks. So the floor is open for questions. You can raise your hand, you can speak out, you can put something in the chat, whatever you'd like. But I want to start. I have several questions. I could ask all the questions for the next hour from you, Cindy, but I won't, so that our participants in the webinar can ask them. But just a quick question to start out. Technology-wise, I read a lot about single cell genomics and spatial genomics going forward. And you mentioned that the Olink technology has a particularly exquisite sensitivity. It can look at very small volumes. And I'm wondering, is it sensitive enough to do maybe not single cell, but small cellular or spatial proteomic type analysis at some point in time?
Yeah, great question. I think this is an area ripe for exploration. I agree that what we're opening up by understanding at a cell-to-cell level, especially the RNA expression within each cell, like fascinating in response and non-responders to immuno-oncology therapy, right? We've got some publications around single cell applications, particularly in that response, non-response area, but where we've played is in the plasma proteome, while the single cell technologies are looking at the tumor microenvironment. And we've got a few publications where the plasma are reflecting signatures in the tumor microenvironment, like tertiary lymphoid structures, these sort of infiltrations of the immune system that show that there's a response to a chemotherapy agent. So far we haven't been able to use our proteomics technology at the single cell level. Certainly there are groups like BioLegend that provide the ability to do cell surface proteins in a sensitive enough space to go into single cell and spatial transcriptomic and DNA.
Okay, thanks. Any questions from the audience? Maybe I will get to ask all my questions. I don't know.
Moderator's discretion, right?
Let me switch then from a technical question to one about your pre-competitive consortium. Who initiated the consortium? Was it the UK Biobank? Was it the pharma partners? Was it Olink? How did the ball get rolling?
Yeah, great question. So this was not us, and in fact, we were told that we had done a good job of staying out of it and just answering questions about what our technology could do, and just trusting if our technology was the right technology for that effort, that it would be chosen. And if it wasn't, then the right technology for that effort would be chosen. So we tried to have good technology that solves a lot of challenges in pharma. But the initiative of creating that UKB Pharma Proteomics project was Chris Whelan, who at the time was at Biogen. Biogen, as I understand it, was part of these sequencing and exome sequencing initiatives within that UK Biobank framework. And those initiatives had been paid for by pharma coming together. And so it was on the heels of that that Chris suggested this. So Lyndon's at Regeneron, Melissa's at Pfizer. Those three really founded this, put together the deck, as I understand it, that was shared among all of the partners that were keen to do this, among all the scientists that were keen to do this. And then within each of those companies, those scientists carried the ball and asked for support to be able to come together to do it. So it was not us, but certainly we have dedicated resources to support that in a scientific way. And I think that's something special I really am proud of about Olink, is to recognize that the science has to lead.
Again, anyone has any questions, now is the time to ask them. Either use the chat box or speak up, raise your hand. Nothing? Well, I'll just keep on rolling through the ones I wrote down while you were speaking.
I love it.
I feel very fortunate to ask all the questions here. But so switching, we're a cohort consortium. And one of the things you mentioned early on was the power of diversity to answer scientific questions. And I know the UK Biobank, for instance, is diverse, but I don't think it's as diverse as the entire galaxy of IHCC consortium members, cohort members.
Totally.
And so I'm wondering, have you seen different effects in different ancestral groups in some of the studies that you've done, either in the consortium or in some of the single experiments that you've talked about, scale-up with the smaller groups? Are you starting to see that diversity be different and give you different answers in different sample sets?
Yeah, great question. And in fact, we'll have an announcement later this week about some more diversity that we'll have housed within the Insight app, showing protein ranges in other ancestral backgrounds other than the Northern Europeans that we have resident in Insight today. So have a look out for those press releases. I can't announce it early. But I think this is a burning question. And then the other question I have in this same vein is, we fully know that there are lots of examples where the ancestral background is controlling protein levels that will alter the risk or be protective, either increase risk or be protective for certain diseases. We've certainly seen that. And so we expect that. To what extent does that persist into old age, where disease progression might take over that genic risk? And we just don't know. I think these are ripe questions that I'm very excited to have this technology accessed to answer. And I think that the UK Biobank Pharma Proteomics project members are very keen to understand diversity as well. I think the UK Biobank is such a unique resource, and it does have some diversity in it, but there's a great opportunity to understand more diversity through working with other cohorts as well. I think there's also great appetite and a keen hunger to understand longitudinal data, much like that Magis et al. Publication I talked about for early cancer detection. Longitudinal sampling will allow us to understand the signatures of disease progression much better, particularly within individuals. And I think that's something those pharma partners have also expressed an interest in understanding. So I think these are all opportunities, perhaps, for the right cohorts.
Ron Ling, you have your hand up. Would you like to ask a question?
Yeah, thank you so much, Cindy, for the nice presentation, which is wonderful. And I'm going to switch the gear from the science to the operation. Actually, the G2MC, GGMC, and we also have the IHCC. Especially for the IHCC, we have over 60 cohorts. Some cohorts from the low middle income country, and some cohorts, the cohort from all 5 continents. Some cohort have the biospecimen, and they have the DNA, they have some other biospecimens. In terms of collaboration, what you can see from the Illumina side, how Illumina can collaborate with the IHCC, G2MC investigators to get those resources for the science.
Yeah.
What do you think?
Yeah. So what I know about Illumina today, so I left Illumina in 2018. I'm at Olink today, but what I still know about them, and I certainly have lots of colleagues that are still there, it's always going to be a very dear company to me, is that they have a population genomics team very interested in exactly this question. So I think that to the extent that these cohorts can be organized and particularly be attractive for industry funding, I think Illumina will also likely be interested in being one of those industry partners to be considered. So it's just what I know about Illumina, but I can't certainly speak for Illumina. I think it's in Illumina's best interest, just like it was in 2014 when I was there leading Consortia. It's in Illumina's best interest for more diversity to be cataloged. I agree with you. And I think this is why I've got such a warmth toward IHCC, and always have, because this ability to sort of draft behind one consortium that's getting access to new technologies and learn those lessons is awesome, and well, I think it's a great position.
Thank you. Yeah, well, I think we are working with them, not me. The whole consortium work with the Illumina. And since we have the enriched resources that can be used for the science. Thank you so much.
Of course. And I think Olink's very, very supportive of, I'm hammering all the time internally when I'm talking to the folks at Olink. They're very aware of this lack of diversity in genetic resources that we want to see rectified. And again, we don't want to see proteomics lag behind that.
I totally agree with you. Thank you.
My pleasure. Thanks so much.
Okay, there's a question in the chat. Let me ask it unless the person that put it there would like to ask it. But the question is, there you go. Would you like to ask the question?
Yeah. Hey, hi. Thanks, Samia Jamar here from Singapore. Thanks for the wonderful talk, Cindy. I mean, it's fascinating. I'm a clinical geneticist, so I look at rare diseases, and I just wanted to ask a tangential question, moving from cohorts to rare diseases. Is there a role for Olink for those undiagnosed cases?
That's a great question.
I believe, yeah.
You go ahead. Sorry, I interrupted you. I just got excited. Go ahead, please finish your question.
No. I'm excited too. As I was listening to your talk, I was like, look, how can I use this technology to help my patients? If it isn't something in the pipeline now, will it be in the future? So yeah.
Such a great question. So I actually work with a group that was very excited about SYNGAP1, which is one of these single gene causes of autism, rare disease, but not as rare as many diseases. And I went to one of their meetings. I actually met the group through a social media anyway. And and they said, oh, you know, we know what Olink is. And in 2018, we actually asked Olink to build an assay for SYNGAP1, which is the protein that is affected within the mutation in the mutated gene. And I said, well, we've got a new product coming out. This was in 2020, at the end of 2020. And I pulled out my Excel and I looked in our planned list of markers, and SYNGAP1 was there. It was because they had asked for it that our R&D team put it into that assay. That was just like, that gave me chills. I couldn't believe it. So they're now, you know, galvanizing, bringing together the group. And the hope is to have a sort of a surrogate endpoint, right? To be able to test potential alternate therapies that are already available on the market to see if they can help their kids, right? So it's such a passion thing. Okay, so that, I wanted to tell that story. So thank you for listening to that. But in the broader sense, right, these groups like Stephen Kingsmore, Project Baby Bear, you know, many groups internationally are looking to improve diagnostic yield, and genetics has given a great advance on that. From what I hear, it's around 30 to 40%. Can proteomics run in conjunction with genomics? Can it improve diagnostic yield? Maybe. That hasn't been tested yet. I think it's a great question that needs to be tested. But what I have even more hope for is that there might be something more than just diagnosis. That, like what the SYNGAP group is doing, there might be the potential to narrow down to therapeutic pathways, potential pathways that are affected based upon the understanding of cases and controls that might be able to be monitored then. It's a ripe area. And, you know, Olink has only been around in this broad look since 2020. That's when I saw the NGS readout. We have so much we could do with this. And this is one of the areas that I'm really passionate about. So thanks for the question. And I don't have any answer for you because I'm hoping that we'll be able to do something in this space.
Perfect. That's great. And if you are looking for guinea pigs, you know, I'm the lead PI for the Singapore Undiagnosed Disease Program. Happy to collaborate with you and see how we can do that.
Well, let's put our heads together and let's see if there's not some way we might get something funded. I would love that. That would be awesome.
Great. One more question in the chat, and it may be time for us to be the last question. Vicky, would you like to ask the question? Would you like me to read it?
Yes, thank you so much for the interesting talk. So I was just wondering, because there's limited expertise in generating and analyzing proteomic data for a lot of cohorts and you know, registries that are being developed in Africa at the moment. And I'm wondering whether there are any plans of a capacity development, you know, wing from either your company or the consortium of pharmaceutical companies to actually ensure that there's more access to the technology.
Yes.
Question.
So it's such a great question. So we have two sides of our products. One is a qPCR readout, which is a very simple instrument about the size of a MiSeq. If you're familiar with MiSeq, it's sort of like a MiSeq, but up on its end. It's just a benchtop instrument where we can read out 92 proteins at a time, and we have 15 different panels of 92 different proteins. That's our target technology, and that was what we, you know, launched the company with. Now I've talked really about the Explore, what we call our Explore technology, and that's on the NGS readout. But that qPCR instrumentation is it doesn't even require anyone to train. It's really a virtual training, pretty simple out of the box. And so the first placement in Africa was in South Africa, not surprising, but I think there's enormous opportunity to bring that technology into different areas in Africa to get access to this technology. Right now, it's part of the Decipher and Diplomics network, and I have the contacts for folks that are running that, and they're very excited about being able to leverage that to characterize diversity. It's not the solution, but it's early stage yet, but I think I think we're very keen to see that done.
Thanks, Sydney. I think that's going to be the end of the questions. And I know Latifa has an announcement she'd like to make about our next GGMC educational webinar series.
Awesome. Thanks so much.
Thank you, Scott. Thank you for moderating this session. Thank you, Sydney, for this insightful presentation. Actually, I learned a lot about this technology for saving lives, as Scott mentioned. Yeah, I put on the chat box the upcoming GGMC educational webinar series. The talk of this webinar is about reflections of biomedical scientists on 4 continents, the importance of mentors, students, collaborators, and interdisciplinarity. And this talk is by our guest speaker, Dr. Jürgen Richard from the Australian Institute of Tropical Health and Medicine. Please mark your calendar for this exciting webinar. It's coming soon, July 27th at 9 a.m., 1 p.m. UTC time. Also, I am going to send you the recording for this presentation, and please don't hesitate to send it to your colleagues or the ones who missed this presentation. At the same time, I will be sending you also the invite for this upcoming meeting. Thank you again for your valuable participation and your questions. Thank you again. And
Thank you. Bye.
Thanks, Latifa.
Thank you. Thank you, Cindy. Thank you again. Thank you, everybody.
My pleasure. Absolute pleasure. Thank you.
Thank you. Bye-bye, everyone.